Hello my dear students, welcome to Biology at Priyajit. In today's lecture we will discuss one of the most important topics in human physiology, the structure of the nephron, the functional unit of the kidneys. If you are preparing for NEET, C.B.Sc, I.Sc, West Bengal Board examination, B.Sc or M.Sc or any kind of university competitive examination, this class will help you understand the basic fundamental structure of nephron in a simple and systematic manner. So let's begin. What is nephron? Basically, the nephron is the structural and functional unit of the kidney, which are the two reddish bean-shaped organs found just above the waist on either side of the spine. They lie behind the abdominal lining, so they are called the retroperitoneal organs. So now it comes into the nephron. The basic functions of nephrons are the filtration of blood, to excrete out the metabolic wastes from our body, reabsorption of useful substances into our body, secretion of unwanted metabolic materials, formation of urine, etc. Each human kidneys contain approximately 1 million nephrons, making them highly efficient blood filtering organs. What are the main parts of a nephron? Each nephron basically consists of two major parts. Number one is renal corpuscle and number two is renal tubule. Let's study each part one by one. Renal capsule. The renal capsule is a renal corpuscle. Renal corpuscle is the site where blood filtration begins. It consists of two components. Number one is glomerulus. The glomerulus is a network of blood capillaries supplied by the afferent arteriole. Here blood is filtered under high pressure. Bowman, second one is the Bowman's capsule or glomerular capsule. Surrounding the glomerulus, there is a double-walled cup-shaped structure called Bowman's capsule or glomerular capsule. Its function is to collect the filtered plasma, which then enters the renal tubule. So remember, glomerulus filters the blood and Bowman capsule collects the filtrate. So this is the image showing the kidney. This is the frontal plane, this is the frontal section of right kidney, and within kidney there is nephron. Within nephron there is glomerulus, and within glomerulus there is a blood supply. We have to know the path of the blood flow. First of all the renal artery, then segmental arteries, then interlobar arteries, arcuate arteries, cortical radiate arteries, then afferent arterioles, which comes into the glomerulus, then glomerular capillary it will form, then it will goes out as afferent arteriole, then it forms the peritubular capillaries, then cortical radiate veins, then arcuate veins, then interlobar veins, and finally renal vein. This is the path of blood flow within the nephron and kidney. Renal tubule. After filtration from the Bowman capsule, the filtrate enters the renal tubule. The tubule has three major parts arranged in one by one sequentially. First one is the proximal convoluted tubule or PCT, which is attached directly to the Bowman's capsule. The word proximal means near the renal corpuscle, while convoluted means it is highly coiled. This PCT is the region where the majority of water, glucose, amino acids and ions are reabsorbed. Secondly, it is the loop of Henle. From the proximal convoluted tubule, the filtrate enters the loop of Henle. The loop has two limbs. Number one is descending limb and the other one is the ascending limb. The descending limb extends from the cortex into the renal medulla. At the bottom, it forms a hairpin bend and returns towards the cortex as the ascending limb. The loop of Henle plays a vital role in concentrating urine by establishing an osmotic gradient in the renal medulla. Third, distal convoluted tubule or DCT. After leaving the loop of Henle, the filtrate then enters into the distal convoluted tubule or DCT. The term here, the distal, means further away from the renal corpuscle. Like the PCT, it is also highly coiled. So it is also called as convoluted tubule. The DCT mainly regulates the final reabsorption and secretion of ions under hormonal control. So for proximal convoluted tubule, Henle's loop, distal convoluted tubule, and several distal convoluted tubules further join toward to form a collecting duct. Many collecting ducts unite to form a papillary duct. Finally, the papillary ducts drain urine into the minor calyces, from where urine flows into the renal pelvis and then into the ureter. Although each kidney contains 1 million nephrons, the number of collecting ducts and papillary duct is much smaller because many nephrons share the same collecting system. Location of different parts. Let's understand where each part is located. The following structures lie in the renal cortex, renal corpuscle, proximal convoluted tubule and distal convoluted tubule. The loop of Henle extends from the cortex into the renal medulla, forms a hairpin turn named as Henle's loop, and returns to the cortex. This arrangement is essential for efficient urine formation. So what are the types of nephrons? Nephrons are basically two types. Number one is cortical nephron. Approximately 80 to 85% of all nephrons are cortical nephrons. Their characteristics include renal corpuscles located in the outer cortex and short loops of Henle. Loops extend only slightly into the other medulla. Blood supply mainly from peritubular capillaries, and these nephrons are primarily responsible for filtration and reabsorption. Second one is the juxtamedullary nephrons. This is the image showing this is the position of the cortical nephron. Most of the portion it is found in the cortical portion of the kidney, and this is juxtamedullary nephron. So the remaining 85% nephrons are cortical nephron, and remaining 15 to 20% are juxtamedullary nephron. Their characteristics are the renal corpuscles located near the corticomedullary junction, very long loops of Henle extending deep into the medulla. Blood supply from both peritubular capillaries and the Vasa recta. The ascending limb has a thin ascending limb followed by a thick ascending limb. This is the thin ascending limb and this is thick ascending limb. These long loops help establish the medullary osmotic gradient. As a result, juxtamedullary nephrons enable the kidneys to produce either very concentrated urine during dehydration or very dilute urine when excess water must be excreted. Understanding the structure of nephron, it is the foundation for learning urine formation, glomerular filtration, tubular reabsorption and tubular secretion under the countercurrent mechanism. If you understand the anatomy clearly, the physiology becomes much easier. If you found this lecture helpful, please like, share and subscribe to Biology at Priyajit. Don't forget to press the bell icon so that you never miss our upcoming lectures on zoology or biology, NEET biology, human physiology and university level science topics. Thank you for watching the video. See you in the next class.